Today we will talk about the backcross. What is backcross? Basically, backcross is simply a breeding experiment which is performed between hybrid organism and one of the parents. So this is simply a breeding experiment. Backcross is a breeding experiment. It is performed between hybrid organism. Backcross is simply a breeding experiment which is performed between hybrid organism and one of the parents. One of the parents. So, what do you mean by backcross? Backcross is simply a breeding experiment which is performed between hybrid organism and one of the parents. We have got a hybrid organism, and then we will just cross this hybrid organism with one of the parents. Then it is called as backcross. For example, if we have got two parents, A and B, then we'll get C. We've got two parents, A and B. Then in backcross, in case of backcross, this is hybrid organism, F1. This is hybrid organism. It is being crossed with one of the parents. So we say that it is a backcross. So backcross is a breeding experiment which is performed between hybrid organism and one of the parents. So for example, we have taken the hybrid organism and is being crossed with one of the parents. So it is a backcross. Remember that backcross is a breeding experiment which is performed between hybrid organism and one of the parents. Now the question arises that why the backcross is being performed. What is the objective of this backcross? This backcross, it increases the traits of the parents in the progeny. So why the backcross is being performed? Backcross is being performed so as to increase the traits of the parents in the progeny. So backcross increases the traits of parents in the progeny. Increase the traits of parents in progeny. So backcross increase the traits of parents in the progeny. For example, how I explain this? We have got a backcross increase the traits of parents in the progeny. For example, we have got a crop plant, and this crop plant is being crossed with wild parental variety so as to get the trait of disease resistance from the wild variety. For example, we have got a crop plant. We have got a crop plant, and this crop plant is being crossed. It is being crossed with the wild parental variety. Why this cross plant is crossed with the wild variety? Because of the fact that this wild parental variety, it contains trait of disease resistance. So as to get the gene for the disease resistance, this crop plant is being crossed with the wild parental variety. So what I mean to say is that why this backcross is being performed. Backcross is being performed, it increases the traits of parents in the progeny. Now we'll explain what the backcross is. Another important thing regarding the backcross is that it is used in horticulture and plant breeding programs, and the objective of there is to increase the traits of parents in the progeny, in the offsprings. And in case the F1 hybrid organism is being crossed with the homozygous recessive parent, at that time backcross becomes test cross. So we can say that backcross also includes test cross. How backcross also includes test cross? If hybrid organism is being crossed with homozygous recessive parent, at that time backcross becomes test cross. This backcross also includes test cross when this hybrid organism is being crossed with homozygous recessive parent. Now explanation for this backcross. We have parents. Suppose we'll take two parents. One is tall pea plant. Suppose we take two parents, tall pea plant and dwarf pea plant. Obviously this is pure, so genotype is capital T, capital T. This is genotype, capital T, capital T. Obviously this is phenotype. This is the genotype of dwarf pea plant. Now gametes, two gametes will be formed. This is capital T, two gametes are formed. One allele will go to one gamete in this case also. So here two gametes are formed. So we have got random fertilization, and what we get? We get F1 generation. For example, this is capital T, small t. So in random fertilization between the gametes, we get F1 generation, first filial generation. So capital T, small t. So tall. Also we will get this one, capital T, small t. So here what we get? This is tall, but this is hybrid. All individuals in the F1 generation are tall, but they are hybrid tall. Now what is backcross? We'll go for the backcross. Backcross is when the F1 hybrid organism is being crossed with one of the parents. For example, we'll cross it with the dominant parent. So we'll cross hybrid F1 hybrid organism is crossed. Backcross is simply when you cross the hybrid organism with one of the parents. So F1 hybrid organism, this one, it is being crossed with one of the parents. We'll take dominant parent. First we'll take dominant parent. So we have got this capital T genotype. genotype is capital T, small t, hybrid organism. Here we have got genotype is dominant parent, capital T, capital T. Obviously, this is genotype. This is phenotype. Then this is phenotype. This is genotype. Then what would be their gamete formation for the fertilization program? We have gamete formation. We have gamete formation. Then capital T, small t. Obviously, capital T gametes. Then by means of Punnett square, we will explain the progeny, back cross progeny. We'll have this one. Oh, these are here male gametes, these are female gametes. So we have capital T, this is small t. Obviously, then here capital T, then another gamete, capital T, and then fertilization, fusion of these gametes. We have capital T, capital T. So capital T, capital T. This one, this one, this one, and this one, capital T, small t. Capital T, capital T, and capital T, small t. So we have back cross progeny. This is back cross progeny. So here we have all, this is tall, this is tall, tall, this is tall. So all tall plants, no dwarf plant, no dwarf pea plant. So in the back cross progeny, what we got, what we obtained only tall plants. Only tall plants. No plant was dwarf or of intermediate size. That means this dominant trait, tallness, tall character, tall trait, it has got inoculated in the progeny. That is why we have obtained only the tall plants in the back cross progeny. This is the result of this back cross. Then we'll perform here we have taken the hybrid organism, and then we have crossed it with the dominant parent. Now in the second cross, we'll take the hybrid organism, and then we will cross it with the homozygous recessive parent. We will see the result. Because I told you that this back cross is simply a cross which is being performed between the hybrid organism and one of the parents. In the first case, we took this dominant parent, and in the second case, we'll take the homozygous recessive parent. So we know that if back cross becomes test cross if hybrid organism is crossed with homozygous recessive organism. Back cross becomes test cross if now when the back cross will become test cross, if the hybrid organism is being crossed with homozygous recessive parent. If a hybrid organism is crossed with homozygous recessive parent, so we have hybrid organism here, F1 hybrid organism. We have that is hybrid organism. It is being crossed with homozygous recessive parent. So we have these are parents. hybrid organism we have capital T, small t, and then homozygous recessive. Homozygous recessive parent we have this small t, small t. This is homozygous recessive parent. Homozygous condition means that if you have got two alleles for a gene and those two alleles are identical, then it is called as homozygous condition or pure. While as hybrid organism or hybrid individual has two dissimilar alleles for the gene located on a pair of homologous chromosomes. So that is a hybrid organism. So this is genotype. Then gametes, gamete formation essential for the fertilization. So gametes are formed here. This is gamete. Gametes are formed here. Gametes. Then we'll perform the cross by means of a Punnett square. Punnett square. We have male gametes, then female gametes. These are being few. There is a union of these male gametes. Female gametes will take capital T, small t as male gametes, and small t as female gametes. So capital T, small t, capital T, small t, capital T. So we have got this capital T, small t, sorry, this capital T, small t, small t, small t, capital T, small t, capital T, small t, and then small t, small t. So we have this capital T, small t, this capital T, small t, then small t, small t, this is small t, small t, this, then we have got here capital T, small t, capital T, small t, and then we have small t and small t, this is small t, small t. It means that what this is the genotype of these individuals, test cross individuals. So this is genotype of this progeny. This is test cross progeny. Yeah, what we called as F2 generation, second filial generation. We have got this is phenotype would be tall here. What is phenotype? Dwarf. So what is phenotype here? This is tall and this is dwarf. So we have got in case of test cross, in case of test cross, in case of test cross, we have got tall and dwarf. Tall is to dwarf. In case of test cross, what we obtain? Tall and dwarf. Tall, how many tall? 1, 2, 2 tall. How many dwarf? 1, 2, 2 dwarf. So what is the test cross ratio? This test cross ratio is 1:1. So this becomes our, this is test cross ratio. 1:1, that is test. I will write it here. Test cross ratio is 1:1 in case of a single character. In case of single character, because we have taken only one character, that is plant height, which has got two contrasting forms, dwarf and recessive. So see the difference here. Here we performed the back cross, where back cross we have performed. Here we back cross we performed the hybrid organism with dominant parent. In the back cross progeny, in the F2 generation, we only obtained tall pea plants. No dwarf plant was present. Then we performed the back cross, where we took this hybrid organism and it was crossed with homozygous recessive parent. And if the back cross involves the homozygous recessive parent, this back cross is then called as test cross. So this hybrid organism and homozygous recessive parent, it becomes the test cross. So hybrid organism genotype, homozygous recessive parent, then these are gametes, then by means of Punnett square, so we obtained tall pea plants as well as dwarf pea plants. So tall, how many tall were present? 2. So 2 tall plants, 2 dwarf plants. So test cross ratio is 1:1. So we can say that in case of single character, test cross ratio is 1:1. That means here only one character was taken into consideration, one trait was taken into consideration, that is plant height, and we got the test cross ratio 1:1. In case 2 characteristics were taken into consideration, at that time test cross ratio becomes 1:1:1:1. So this back cross is simply a cross where we perform, this is being performed between the hybrid organism and one of the parents. This increases, what is the objective of this back cross? It increases the traits of parents in the offspring. And if back cross is being performed with homozygous recessive parent, then back cross becomes test cross. So this was all about the back cross. Hope you have understood. Thanks.